All right, are there any questions? So I will talk about the exam towards the end of the lecture, okay, that we have on Thursday. So but now let's just continue with the material. So any questions from last time? So towards the end of the lecture, I was trying to talk about, last time I was trying to talk about sulfur assimilation. And I mentioned that sulfur is going to enter through the plant roots as the sulfate ion, right? And it's going to be drawn in to the plant, through a hydrogen ion, right, proton, sulfate symporter, right? So sulfate ion absorbed through H plus SO4 2 minus symporter, right? And we discussed this before, right? When you have secondary active transport, right, and both the molecules are being transported in the same direction, right? That's called symport, right? So this is how sulfur is going to be absorbed, and then if sulfur is absorbed into the plant, it is usually converted into an amino acid that contains sulfur, right? And we already know cysteine has got those disulfide bonds, right? So this is going to get converted into cysteine, right? And then this can get converted into another amino acid that contains sulfur, which is called methionine, right? It's one of the 20 amino acids that we have listed right at the beginning of this class, right? So that's sulfur assimilation, right? Then the next thing I wanted to talk about was phosphate assimilation. And again, here also phosphate is going to be assimilated as the phosphate ion, right? And it's going to be carried out, the assimilation is going to be carried out through proton phosphate ion symporter, okay? So phosphate in the soil solution is readily transported into plant roots via a hydrogen ion, HPO4 2 minus symporter. And what do you need phosphates for? What kind of molecules in plants are going to have phosphates? Go ahead. Okay, ATP, what else? Yeah. You have the sugar phosphate backbone and so on, okay? So now you know enough. Having taken this class, you know that phosphates are needed, okay? Although this material is not there on this test, okay? But at least you're retaining something. You already know about that stuff, okay? All right. Then the last item, then yeah, so DNA, RNA, and so on, right? Then phospholipids, right? DNA, RNA, phospholipids. So these are the molecules, some of the molecules where the phosphate is going to end up, right? Then next, I want to talk about cation assimilation, right? So cations are basically positively charged ions, right? You know that, like sodium, potassium, calcium, these are all cations, right? So cations actually form what are called, so cations form coordination bonds. And electrostatic bonds. With carbon compounds. Now electrostatic bonds, you're all familiar with that, right? Attraction between positive and negative charges. What are coordination bonds, right? Well, these are not covalent bonds, but you might have oxygen and nitrogen, right? That might actually uh, donate their unshared electrons to the cation. Like when oxygen and nitrogen, they form bonds, covalent bonds, you have sharing of electrons, but there might be unshared electrons that these guys could donate to the cations, and those are called coordination bonds, right? So in coordination bonds, several oxygen or nitrogen atoms donate unshared electrons to the cations. So let's talk a little bit about one particular cation. Okay? Iron is absorbed from the soil by the roots of the plant. Okay? So in order to absorb the iron, right, the plant has to do some extra work because sometimes the iron is present as ferric ion, right, which is not readily absorbed, right. So the roots have to do extra work. So roots modify the surrounding soil to acquire iron. And they do this in at least a couple of ways. Number one is soil acidification. And how can the roots do that? How can the roots make the surrounding soil more acidic? By doing what? By pumping what? Pumping what? Protons, yeah. Yeah. So soil acidification, which increases the, the solubility of ferric iron. So Fe3 plus, right? That's ferric iron, followed by the reduction of ferric iron, so Fe3 plus, to the more soluble ferrous iron. So more soluble Fe2 plus iron. And then the second thing that they can do is the roots can also release compounds. So release of compounds that form stable soluble complexes with iron. And these are called iron chelators. Right? And the book has more details on those, at an earlier chapter, right? But I'm not going to get into that detail, but they'll put some molecules in that'll make the iron, render the iron more soluble so that the plant can absorb it, right? All right, that finishes our discussion of the chapter on absorption of those nutrients. Okay, like we did nitrogen fixation and things like that last time. Okay, that completes this chapter. So any questions so far? Otherwise, we are going to move on to the next chapter, right? And again, I'll talk more about the exam later on, Thursday's exam, but the exam is going to cover up to whatever we finished last Tuesday, right? So today's material, I mean, I've covered a little bit, but you don't have to worry about that. So now let's move on to the next chapter, which is chapter 14. And chapter is devoted to cell walls, structure, formation, and expansion. And as in all the chapters that I'm covering, I will try to just give you the big picture, right? So it doesn't get too boring, too much of memorization, right? You're not going to be tested on this, but I hope at least you've learned enough stuff in this class to feel some interest in learning whatever we're covering now, okay? Now, as I indicated earlier, right, an important difference between plants and animals is that the plant cells are enclosed inside a rigid cell wall, right? So plant cells enclosed by a rigid cell wall. The cell wall provides mechanical protection to the cell and allows it to withstand large turgor pressure. Right, we've talked about this before, right? Also, when water is being drawn up the xylem, right, there is tension, right, because there's a transpiration stream going on. So this cell wall makes sure that the thing doesn't collapse, okay, when there is tension or negative pressure. So resists collapse in response to negative pressure in the xylem. And the negative pressure arises from the transpirational water flow. Now the presence of the cell wall, right, is going to prevent cell sliding, right, because the cells cannot move around freely because you have the limiting cell wall. So it prevents a number of things, right? So prevents cell sliding, then slippage is going to hold things in place, right? So slippage, motility, that means the mobility of the cells, right? And this situation is different from cells in animals, right? Situation different from that in animal cells. That's why, as I mentioned earlier, in the case of plant cells, all right, the neighboring cells will have to have a common lineage, okay? You cannot have a cell that is really far away, and then that's coming from the same lineage as some cell that is here, unless everything originates from the initial zygote, the fertilized cell, Now, in the case of plant cells, you have two kinds of cell walls, and I've talked about this before, but I'm going to repeat it again. So you have something called the primary cell wall, which is basically laid out as the cell grows in size, right? So it has to be thinner and, it has to be kind of expansible, right? So thinner, and this grows as the cell expands, right? And then you have the secondary cell wall that is inside the primary cell wall, all right? Once the cell has stopped growing, then the secondary cell wall is laid out. That's going to be a lot harder, right? So secondary cell wall is deposited between primary cell wall and the plasma membrane. So, and plasma membrane. Once cell growth has stopped. Right? And in the case of xylem tissue, because those walls have to be even more reinforced, right? There's something called lignin that gets deposited in it. right? And there are multiple layers to make it really hard and so that it can withstand the water pressure. So xylem tissue has multi-layered secondary walls that are strengthened and waterproofed by lignin. And these walls are said to be lignified. And again, we don't have time to get into the detailed structure, chemical composition of lignin and so on, right? But I'm just mentioning that. Now, what are cell walls made of? What do you think cell walls are made of? Cell, yeah. So it's basically sugars, all right? Polysaccharides of sugars, right? So whatever we learned in the first chapter, right, maybe it seems like ages ago, well, all that is relevant right now. So cell walls are made of different polysaccharides. And the composition will obviously differ between the primary and the secondary cell walls because one of them has to allow expansion, right? The other one doesn't. The other one has to, basically reinforce rigidity, right? So the composition differs between primary and secondary cell walls. All right, so let's just introduce some nomenclature here, right? So we all agree that the cell wall is going to be made up of polysaccharides, right? Polysaccharides mean sugars that are linked together, right? Remember, and they're linked together in what kind of reaction? Condensation reaction. Good. Yeah. Not showing up on this exam, but at least you retain something, which is good. So nomenclature. I mean, and different polysaccharides have different names, right? So glucan, right, is basically a polymer that is made up of glucose units that are linked together. Polymer made up of glucose units linked together, right? Then a galactan, right? What do you think a galactan is going to be? Galactose units linked together, right? So galactose units, right? Then you have xylan is made up of another sugar, right? Xylose units that are linked together, right? And again, all of these are synthesized in condensation reactions, right? So xylose units linked together. Similarly, you have mannans. These are mannose units linked together. Then finally, if somebody says is talking about glycan, this is generally just another name for a polysaccharide. Right? It's not any particular set of sugars. So this is a synonym for a polysaccharide. And then you may have a situation where there is a backbone that is made of one particular sugar, and then you have branches that are made of other sugars, right? So the terminology that is usually used to describe, not always, right, but in many cases, is something like this. If somebody says this is a xyloglucan, right, that means the backbone is made of glucose units linked together, and then the side chains are made of xylose, right? So glucan backbone with xylose sugars. Then another example could be arabinoxylan, right? It will have a backbone that is xylan made of xylose, right? Xylan backbone. and then the side chains, right, will be arabinose, right, with arabinose side chains. Now, if you look at the cell wall, right, there are three kinds of polymers that are involved in the cell wall. Right? One of them you said already is cellulose, right? Another one is called pectin, and another one is hemicellulose, right? So three kinds of polymers. One of them is cellulose, and this is made up of what are called beta 1,4 linked glucan. Again, glucan is basically glucose units linked together. Beta is basically, it's telling you it is an optical, some kind of an optical isomer, right? 1,4 is carbon 1 is connected to carbon 4 of the next one, right? I'm just trying to explain the terminology. We don't need to know the details, but so it's made up of beta 1,4 linked. It's a beta 1,4 linked glucan. It is insoluble in water, and it has a high tensile strength. Then if you look at pectin, so pectin is something that is hydrophilic. It's a gel forming polysaccharides rich in acidic sugar residues. And again, if you look at the book, they'll give you the detailed structure of all these molecules. Right? So hydrophilic gel forming polysaccharides rich in acidic sugar residues. Yeah, I believe the skin of apple and all that contains pectin. Right? All right, and then the third kind of polymer that you have is called hemicellulose. Right? So hemi means half, right? So this has got polysaccharides with beta 1,4 linked backbones requiring strong extractant. So if you're trying to dissolve this out, it's not easy, right? Requiring strong extractant. For example, maybe like 1 to 4 molar sodium hydroxide. To be solubilized. So of these three, only pectin is the one that is soluble in water, right? So if you look at the primary, so we've talked about primary cell wall and secondary cell wall, right? So primary cell wall will mainly have hemicellulose and it will have pectin, because remember the primary cell wall has to have the flexibility, right? So primary cell wall, so primary cell wall is made of pectin, mostly of pectin and hemicellulose. And then secondary cell wall is made of cellulose, hemicellulose, and different amounts of lignin. because the lignin is going to make it hard and waterproofed, right? Now, when the cell wants to expand, right? Cell wants to expand, then what happens? The wall has to loosen up, right? So there are proteins that are called expansins, right? That will loosen up the cell wall so that the cell can expand when needed, right? So expansins, these are proteins. So expansins catalyze the pH dependent extension and stress relaxation of cell walls. And that's how the cell wall can grow, right, if needed. And cell walls extend much faster at acidic pH than at neutral pH. Right? So you can well guess that if the cell wall needs to expand, right, the cell will find a mechanism to pump protons, right, into the apoplastic space, right. That will raise the pH, then it will become expansible, right. Okay, any questions? And all this stuff is important, all right? I mean, we don't have to know the details 100% because plant water use is pretty important. Remember, I pointed out before that in order to do photosynthesis, for every molecule of carbon dioxide that you take in, right, you expel how many molecules of water? 400 molecules of water, okay? So if we can drop that down to 50 molecules or something, that's a big deal, right? Especially in Texas. Not today. Today it's raining and all that, but in these dry summers you want to grow plants, okay, with less water, and that's a grand challenge. You want to produce food for a lot more people on the limited amount of land using less resources. Right? That's really a grand challenge, right? So that's why it's better to have some idea about these things. Although you are going to work only on the signaling aspects, right, or engineering aspects, but at least it's good to have some domain knowledge, right? And that's essentially what I'm trying to impart in this course. All right, any questions? So if there are no questions, then we move on to the next chapter, which is about signals and signal transduction, right? So chapter 15. And this is the chapter where you'll find that all the plant genomics and all that links up with the animal genomics, whatever we have already covered. All right. Same methods, microarrays, all right. Next generation sequencing, all these things work here also, all right. And you'll get the signals. The only difference is that you're going to be working with plant cells, all right. So chapter 15 is on signals and signal transduction. Now, elaborate signaling mechanisms, right, are involved in plant development. Even in the case of animals, we have not covered that in detail, right? But even in the case of animals, it is the signaling, right? Like you think about it, a single fertilized egg or a zygote develops into an adult human being, right? With about 100 trillion cells, okay, with the right kind of tissues and everything. All that is achieved basically by turning on the right genes at the right location, right? And at the right time, so that you don't get abnormalities, right? So plants also the same kind of thing has to happen. So elaborate signaling mechanisms are involved in plant development. And I give you a few examples, all right? Phototropism. That means the plant has got a tendency to grow towards light, right? Most plants, right? And we have seen that, right? If you keep a plant inside a house where, near the window, right, it'll try to grow towards the light, right? Phototropism. Then gravitropism. Like the root of a plant is going to go down, right? It'll go in the direction of gravity. So gravitropism. These don't happen automatically. There is signal transduction involved, right? There are signaling pathways, gravitropism, etc. And we will discuss some of these in a little bit more detail, right? So signal receptors located throughout the plant plant cell, right? They are going to take those signals either coming from outside, like maybe sunlight, right? Or some development signals, like something needs to grow, the neighboring cells are telling the cell to divide, right? And then they are going to produce the response, right? So signal receptors located throughout the plant cell to respond to environmental and developmental stimuli. Now we have seen in the case of animal cells, all right, we have seen the role of receptor like kinases, all right. On the cell membrane, all right, you have a receptor, some molecule comes and binds, that produces signaling inside, right. We've talked about the MAP kinase pathway, then we talked about how breakdown in that signaling can lead to cancer and so on, right. So in the case of plants also you have these receptor like kinases. So role of receptor like kinases. Again, what does the kinase do? Adds a phosphate group. Yeah, phosphatase will take off a phosphate group. So you'll have receptor like kinases on the cell membrane, right? That'll produce some signal transduction inside, right? And again, I mean, the signal should not die down as it is being transmitted. So you might need amplification of the signal, right? It's just like before you do like a radio broadcast or something, you amplify the signal and then you send it over, okay? So intracellular amplification of signals is needed. And in fact, a particular example where you do have intracellular application of amplification of signals is the MAPK signaling cascade, which we also see in animals. So, right, all eukaryotes have that. So mitogen activated protein kinase signaling, right? So MAPK signaling cascade. So you basically have serial phosphorylation one after the other to amplify the signal, right? So you have a situation like this, and we have already seen that in the case of animals, all right? So you have a receptor on the cell membrane, right? Some molecule comes and binds here. The ligand will bind the receptor. That will produce some signaling, some kinase activity. So you have maybe MAP3K, right? Something gets phosphorylated. Then this one in turn goes and phosphorylates something else, MAP2K, right? And then that might phosphorylate another molecule inside the cell, like MAPK, right? And this could lead to the transcription of some genes, right, that are needed for the, let's say, for the cell to multiply. So that's one example, MAPK signaling cascade. In addition to that, inside the cell you have second messengers. And this is just not for plant cells, even for animal cells that is true, right? So examples of second messengers are the calcium ion concentration, right? That is an important second messenger, right? It is going to transmit signal somewhere, right? So calcium ion concentration. Even in an animal cell, in a fertilized egg, right, for an animal, for that to develop, for the process of cell division and all that to start, the cytosolic calcium concentration has to go up. Right. We didn't discuss all those things, but so it's no different in the case of plant cells. So calcium ion is one of them. Another one is pH, which is basically the hydrogen ion concentration. Then reactive oxygen species, right? So we have encountered reactive oxygen species in the context of photosynthesis, right? Respiration and so on, right? So when you have like things like hydrogen peroxide being produced, right, singlet oxygen atom and all that, so these guys are produced, right, and these can be secondary messengers. So reactive oxygen species, like you hear about people taking antioxidants to prevent aging and all that, basically they're trying to reverse the effect of these reactive oxygen species, all right. So ROS. then you can have lipid signaling molecules. For example, diacetyl glycerol. So we've talked about triglycerides, okay? Triglycerides are basically triacyl glycerols. There are 3 acyl groups, right? So from that you can get diacyl glycerol by breaking one of the bonds, right? Right? And that molecule can be a signaling molecule. There are different variations of that. I'm not discussing those here, but I mean many different second messenger molecules are possible. So these result from different lipases. What is a lipase? Anybody? No, no, no. Huh? It what? Breaks down lipids, you know. You hear about lipases, it basically break down lipids. Okay, so it might take a fatty acid off, or it might take the phospholipid off, or whatever, right? So it results from different lipases. See, if it synthesizes something, it will be called a synthase. Okay, if it breaks down something, it's going to be called something ase. Okay. All right. Now, what are the signals that the plants respond to? Okay, there can be sunlight, gravity, and so on. Right. In addition to that, plants also have many hormones. Like we have hormones like insulin and so on. Okay. Plants have many hormones too, right? That are important. And so we will discuss these hormones again, not in a whole lot of detail, but at least to give you some idea. So plant hormones. So these are like internally generated signals in the plant, right? Not coming from the external environment. So examples of plant hormones are auxin, right? That is the first plant hormone to be discovered. This is the enzyme. This is the hormone that causes bending, right? In response to light, right? The plant moves towards the window. Why does that happen, right? Because at the tip of the plant, auxin is produced, right? And that goes down. On the shady side, it is going to cause the cells to multiply a lot faster, so that it will become longer, right? So the growth on the shady side will be longer than the growth on the side that is receiving the light, and as a result, the plant will bend in that direction. So there is signal transduction involved. Nothing happens automatically, right? So auxin, it causes bending, and there, auxin causes a lot of different things, right? Because things have to grow. Like if you have a seed that is developing from a zygote, right? All the cells have to grow in a proper orderly fashion, right? The flow of auxin is what modulates that, or at least one of the important factors that modulates that. So auxin causes bending in the direction of light. And auxin is produced by the shoots of the plant, right? So produced by the shoots, but it also travels to the roots, right? And auxin is, the chemical composition of auxin is what is called indole 3-acetic acid. And if you look in the book, they'll give you the formula, right? We don't need to know that. We just need to know that auxin is basically like a growth hormone, right? That will make the plant bend in the direction of light. Because then on the shaded side, right, there's auxin that, and people have done experiments because it's produced from the tip of the plant. Like if you go and block the path from the tip of the plant, right, to the base, then even when you have light, it doesn't bend. Okay, so those experiments, biologists have already carried out. Right? Another plant hormone is what is called gibberellin, and this one promotes stem growth. And in this context, I should mention a disease that is called the foolish seedling disease, right? Because if you give a plant too much of gibberellin, right, the shoot will become really large, right? So in rice plants, it has happened that there is a fungus that produces gibberellin, right? So if you expose this plant to that fungus, excess gibberellin is produced, the shoots become too tall, and then they tip over, right? So the yield is not there, okay, because it tips over. So this is called a foolish seedling disease, right? So promote stem growth. So we should also mention this foolish seedling disease of rice. So it's called foolish seedling because it went up and now it's not producing anything, right? So rice, when infected with fungi that produce gibberellin, too much of gibberellin. Okay. All right, any questions? Now, in this context, I should also mention, how many of you have heard of Norman Borlaug? Anybody heard? Yeah. In what context? Go ahead. Yeah, yeah. Huh? Yes, he's the father of the Green Revolution, and he got the Nobel Peace Prize, right? For doing that, and he used to be a distinguished professor here at Texas A&M. There is actually a center that is called the Borlaug Center. He used to be on campus until I think maybe 3 or 4 years ago, when he passed away at the age of I think 95, right? So, so Norman Borlaug, he's credited with saving more human lives, all right, in the last century than anybody else. He saved like 1 billion lives, all right. He's a hero of people like Bill Gates, okay. Now, what did he do, all right? Basically, he produced a different variety of seed, okay, where the plant is not going to grow that tall, okay. It's going to be shorter, so you can fertilize it. You can fertilize it more and increase the yield. Otherwise, the problem is if it grows too tall, you're going to flip over, right? But that allowed him to save like 1 billion lives in the last century. Okay. So what we are talking about here is not irrelevant stuff. It can make a difference. It is a difficult thing to do, right? But it can be done, right? Norman Borlaug has already shown it. And I don't know which precise hormone or whatever he tinkered with, But we have this center here, and I was talking to the co-director of that center, right? Who's from AgriLife, and he explained this to me that basically when the plant grows shorter, then you can fertilize it and increase the yield. Then another plant hormone is called cytokinin. And this is produced by the roots of the plant. So cytokinins, they promote cell division. And they are produced by the roots. Then another plant hormone. I think there is a total of 9 plant hormones. And again, each one of these hormones has got its own signaling pathway. But the pathways interact with each other, right? So it's going to be a multivariate interaction that is going to produce the final phenotypic response. That's where the engineers come in, okay? Because in engineering, we don't look at things one at a time. We look at the overall interaction and try to predict what is going to happen, right? And that's where some of you guys will find, topics to do your master's, PhD, and so on, okay? So another hormone is ethylene. And does anybody know what ethylene can do? Somebody might know, right? What can ethylene be used for? What? It can be used to do what? For plants? Anybody? Ripen fruits, yes, yeah. So this stuff is really relating to whatever you know from everyday life, okay, yeah. You take bananas, you expose them to ethylene, it's going to ripen, okay. So ethylene promotes, and the fruit ripening doesn't happen automatically. When you put in ethylene over there, there's some signal transduction inside, okay, that is causing some molecules to act on something else, and then you're seeing the phenotype of ripening. So promotes fruit ripening. Then leaf and flower senescence. Senescence means growing old, right? If the fruit is ripening, it's time for the leaf and the flower and all to basically grow old and die, okay, and fall off. Then the next hormone is what is called abscisic acid. Or it's abbreviated as ABA, right? This regulates seed maturation and stomatal closure in response to water stress. So we have already looked at stomatal opening in response to something else. What did we look at? No, pressure will be this, but what was the primary stimulus that was driving it? Light, yeah. Yeah, blue light, right, was driving stomatal opening. But stomatal opening or closure, it can be driven by other things. If there is water stress, not enough water, the stomata better close so that the plant doesn't get desiccated. Okay? So the abscisic acid pathway is the one that modulates that. Then there is another hormone, another class of hormones that are called brasinosteroids. These regulate photomorphogenesis. That means changing, right, the form in response to light, okay, because you need light when the plant grows, okay, then it produces flowers, seeds, then dies and so on, okay. So this is photomorphogenesis, changes that are brought about in form by light, right. So brasinosteroids regulate photomorphogenesis, then germination and other developmental processes. So other developmental processes. Then the next hormone that you have, they're called strigolactones. These suppress see, when a plant is growing, okay, is it going to grow more upwards or is it going to produce lateral branches, right? Again, that is dependent on signaling and hormones, right? So strigolactones basically suppress branching and promote rhizosphere interactions. So the region surrounding the root, the plant root where the root is interacting with bacteria, right, and fungi and so on, that's the rhizosphere, right? So this hormone is going to promote interactions over there. So, so suppress branching and promote rhizosphere interactions with fungi surrounding root, etc. Right? Now all these hormones, right, I have talked about 9 different hormones, 9 different plant hormones, right? All these have to be synthesized, okay? And there are pathways for synthesizing these hormones in plants. I'm just going to talk about 1 or 2 of them, and again, just catch them, right? There are different steps that are involved, right? So hormone synthesis, and the book has details on all of them, right? But again, we are not trying to become plant biologists, so we don't have time to go and... But again, if you're focusing on a particular pathway for your research, you're going to use signal processing methods to figure out, how the abscisic acid pathway affects stomatal closure. You have to go and focus on that particular pathway, learn whatever is there in the book, search the literature, see what is available and all that, and that way you come up with a good problem that you can work on. But you need more domain knowledge over there. Okay. So, and it's impossible for me to cover all those things in the class because I cannot work on all of these different pathways, right? It's up to individual students to pick and choose, right? So, if you look at hormone synthesis, auxin is synthesized from indole 3-pyruvate. And where did we see pyruvate? Where did we see pyruvate when we were discussing what? Glycolysis, yeah. So glycolysis, TCA cycle and so on. So auxin is synthesized from indole 3-pyruvate through the action of the yucca gene product. And again, I'm just mentioning this as an example. I don't know what the structure of the yucca gene product is, but I just mentioned that for each one of them, and there will be different steps in those pathways. Right? Then another example is, for example, strigolactone is synthesized from beta carotene, and there would be a pathway for synthesizing that. Again, I mean, this is very, very sketchy. I don't have time to go into the details of that. Okay, but each individual person, if you're going to do research related to agri-life applications, you would have to pick up a particular pathway, see what is done, right? What more needs to be done, and my own opinion, assessment is that not much is done. In fact, in this chapter in the book on plant physiology and development, right, is not an engineer, but a biologist mentioned, and I'll give you the page number, right, where they say that you need systems biology, right, because they cannot figure out, all the different, because the pathways interact with each other, right, and you are ultimately interested in controlling the phenotype, right. So not a whole lot has been done. Some things are known, like whatever I'm teaching here, but others are not. Right? Now let's talk about signal transmission, right? Like you're producing all these hormones. Are they going to be utilized in the cell where they are produced, or are they going to go over distances and then get utilized, right? So those are also issues that we have to look at, right? And I can give you the big picture here. So signal transmission. Hormones and even other signals also, right? Now, if you're looking at hormones, all right, you can have three possibilities. One would be endocrine, right, where the site of action of the hormone is different, is far away from the site where the hormone is being produced, right? And we have endocrine systems, right? So here the site of action, animals have endocrine systems, so site of action, distant from site of synthesis. Paracrine means the site of action would be the neighboring cells, right? So paracrine hormones, they act on cells adjacent to site of synthesis. And then the third one is autocrine. That means what? Make a guess. They function in the same cell where they are synthesized. Okay? Function in the same cell where synthesized. Now animals, like us, all right, we have endocrine systems, all right, because we have a very, very fast moving circulatory system, right? So the hormones can travel all over the body, right, through the blood vessels, right? So animals have endocrine systems. So transportation is not an issue. Have endocrine hormones easily distributed over long distances using the circulatory system. Most plant hormones are of the paracrine type. Another thing to note is that in the case of animal hormones, all right, Many of them are of the activating type, right? So the hormone will go and activate the pathway. In the case of plant hormones, it is mostly removing the repressor for a pathway, right? So most plant hormonal signaling pathways involve ultimately deactivating a repressor molecule. So this will allow a much faster response, right? So, and the repressor and the activity deactivation could be carried out, let's say, through a kinase or a phosphatase, right? There could be other mechanisms too. So, example, so kinase slash phosphatase activity. And this allows for a much faster response. Because you don't have to carry out any synthesis from scratch. Because if you're trying to generate a new protein or something, you have to do transcription, translation, and so on. Right? Here the protein is already there. Okay, you just went and deactivated it by putting in a phosphate group or removing a phosphate group. Right? So no synthesis. So no synthesis from scratch involved for the target molecule. So this really speeds up the response. And this is important because, see, if an animal, suppose the conditions become adversarial here, okay, let's say the temperature goes up in the immediate neighborhood, okay, what is the animal going to do? It'll move, yeah, it'll run. Plants cannot move, right? They're sessile organisms, so they have to be able to respond quickly, right? So, so this speeds up the response, which is important for ensuring the survivability of an immobile organism in a hostile environment. In animals, many of the signaling pathways are positively regulated. And I give you just one example, all right? Because for each of these hormones, okay, you'll have a different pathway, right? So I give you an example of one pathway, and the others are discussed in the book. And again, as I said before, everybody doesn't need to go and become an expert in every pathway, right? Unless you know that particular pathway is of interest to you, you're going to do theoretical work on that. So I give you just an example, one of the easier ones, the ethylene signaling pathway. So let's say that the ethylene gas has been produced in response to some external conditions. Ethylene gas has been produced in the endoplasmic reticulum, right? So ethylene gas has been produced in this endoplasmic reticulum. So this is the endoplasmic And on the membrane or of this endoplasmic reticulum, there are ethylene receptors that are located, right? So these are ethylene receptors. And so you have ethylene gas that is produced and it comes and binds that receptor. So ethylene. So once this binding takes place, right, there is signal transduction, right, inside the cell. So there will be signal transduction, and there are many steps that take place here, right? I'm not going to fill those in. So this is going to causes signaling inside the cytosol, right? And then finally what happens is that some molecule is released that enters the nucleus, right? So let's say this is the nucleus over here, the nucleus of the cell. So it causes some signaling inside the cytosol. The net result is that some molecule released to go through the nuclear envelope, right? Now the molecule that goes in, right, it is going to release some brake, right, on the synthesis of ethylene's response genes, right? So that molecule, so the molecule that goes in, so this molecule releases the brake on transcription of ethylene responsive genes. Alright? So maybe it goes and releases the promoter region, something else was blocking there, right? So then this will imply that ethylene responsive genes are now produced, they're transcribed, translated, and you have the response that's supposed to take place, right? So it implies ethylene responsive genes are produced. Then you'll get the protein and so on, and some of them could be even transcription factors, right? But it'll get the procedure going, right? All right, any questions? Now plants have evolved mechanisms for switching off or attenuating signaling responses. Right? Because the response should take place only when it is necessary. Okay, once let's say in the case of ethylene, once the ethylene response has taken place, right? Then this ethylene should no longer be producing more of those genes, right? So there has to be a mechanism for doing that. Okay, so one of those could be feedback inhibition, right? Where the final response is negatively regulating the original stimulus. The example of a feedback control system, all right? Or you could have ethylene flowing out of the cell. Right, there could be efflux, there could be transporters that are driving it out so that once the necessary action is over, the activity does not continue. Right? So the mechanisms could be like feedback inhibition, then efflux, etc. Right? Then the response to these different hormones, right, is also usually tissue specific. So response is often tissue specific. For example, auxin is going to stimulate growth in shoots, right, above ground portion, but it basically suppresses growth in the roots, right. So auxin stimulates growth in stem, but inhibits growth in roots. Also, as I have been emphasizing, there's a lot of crosstalk that goes on between different pathways, right? So crosstalk between pathways. So these signaling pathways do not act in isolation, right? So they interact with each other, and therefore there is a need for a multivariate analysis of these different pathways, right? So there is a need for this area of so-called systems biology, right? And I refer you to, let's see, page 443 of this book, right? So if you don't believe me, go and look up the book, page 443. You will see the biologist over there is basically begging for systems biology to come in, okay? But they realize that there is a need for that, right? So it's not something that I have invented, because when I was going to teach this class, I kind of started from scratch. A lot of the stuff was boring and all that. Everything didn't fall in place, right? But earlier this summer, I suddenly, came across that, and then I said, okay, I'm doing the right thing, for myself as well as for the students. Right? All right, any questions? So that finishes our discussion of chapter 15, right? I'm now ready to move on to chapter 16, right? So we have talked about general signal transduction. Now in chapter 16, we will focus on signals from sunlight, okay? Where the stimulus is sunlight that is driving the signal transduction, right? So chapter 16 is signals from sunlight. So in addition to enabling photosynthesis, sunlight plays an important role as a signal for plant growth and movement. And I give you examples, right? Several examples actually. Example, the solar tracking of leaves, right, of some plants. We talked about this before, right? As the sun moves across the sky, the leaves also change their orientation. So solar tracking of leaves. Then photomorphogenesis. That means changing form, right, in response to sunlight, right? And examples of this are inhibiting stem elongation, in growing seedlings. See, when you plant a seed underneath the ground, okay, there's germination that takes place, right? So initially that seedling is not getting any light. That time the stem grows really fast, right, incredibly fast, maybe millimeters an hour or something like that. As soon as it hits the ground, right, then it sees light, right? Then the growth rate slows down, okay, because now it's going to focus on doing other things, okay, not just shooting upward just to get out of the ground, right? So that is affected by light. So then phototropism, that is the tendency of plants to grow towards light. So growing towards light. Then photoperiodism. So the plants can distinguish between seasons, right? Like because in summer the days are longer, right? and the nights are shorter, right? In winter around this time the nights have suddenly become longer. So that's how plants figure out, okay? Is it spring or is it autumn, right? The length of the night, right, basically dictates that. And I will talk about that in a later chapter, right? So photoperiodism is basically distinction between the different seasons. So distinguish between seasons by using the changing lengths of days and flower accordingly. So the signals from sunlight, they are processed by what are called photoreceptors, right? So just like you have on the cell membrane, you have a receptor, right? We talked about the ethylene receptor. Same way for light, there is photoreceptor, right? So unlike the photosynthetic pigments, right, like chlorophyll and all that, they will absorb light at a particular wavelength and release light at some other wavelength, right? And they will cause all those photochemical reactions, right? Photoreceptors will basically absorb light and then use the energy for signal transduction, right? So unlike photosynthetic pigments, photoreceptors absorb a photon of a given wavelength and use the energy as a signal to initiate a photoresponse. Most known photoreceptors consist of a protein plus a light absorbing chromophore. And if you look at a photoreceptor, right, this is just some terminology. If you have the protein plus the chromophore, that is called the holoprotein, right? Without the chromophore, it is called the apoprotein. So just some terminology. Again, nothing to memorize because this is not going to show up on the test. So holoprotein is protein plus the chromophore, and apoprotein is protein minus the chromophore, right? Now the different kinds of photoreceptors that we will talk about, right, are the following. So photoreceptors. The first set of photoreceptors that we talk about, those are called the phytochromes, right? Phytochromes, right? Uh, these phytochromes, they are responsible for plant photomorphogenesis, all kinds of plant photomorphogenesis, and they absorb mainly red and far red light. Okay, because if you look at the spectrum, you're going from red to blue, right? Blue and then violet, right? So these phytochromes, they absorb mainly red and far red light. Then the next group of photoreceptors are what are called the cryptochromes. And these photoreceptors, they are also involved in plant photomorphogenesis, and they absorb blue light, right? Photomorphogenesis, and they absorb blue light. The next group of photoreceptors are called phototropins. And they induce the responses like phototropism, that means growing towards light. Then chloroplast movement, when there is too intense sunlight, all right, then the things move to the side, the chloroplast, because it doesn't want to absorb too much of light, right? So that's carried out by phototropins. So chloroplast movements. Then they also regulate stomatal opening. And these phototropins are activated by blue light. We already talked about this, how the stomata open when you have blue light, okay? Remember that ATPase is going to do proton pumping, potassium ions get in, chloride ions get in, water flows in because of the reduction of water potential, right? And then the stomata open, right, in the guard cells, right? So that's basically activated by phototropins. And then the last one is UVR8. This is only recently discovered, and this one is responsible for response to ultraviolet light B, UVB. Okay, and this one we are not going to worry about. I'm mainly concerned about red light, far red, and blue. Okay. Now the plant response to light is determined by a couple of factors, right? So plant response to light depends on, number one, the fluence, and number two, the irradiance. So fluence is basically the light that is falling per unit area. So light per unit area. So light per unit area. And irradiance is light per unit area per unit time. That means per second. Okay, so that's like the intensity, right? So per unit area per unit time. And both of these factors can be important. Okay, it might be important to give a lot of energy over a short period of time, right? Right? Or in some other situation, it might be important. It doesn't matter. You know, the total amount of energy. It doesn't matter over how much time it is spread out. All right. So first, let's talk about phytochromes. So phytochromes are photoreceptors that absorb red and far red light. Okay, and they have two forms, right? PR, right, is the phytochrome that is going to absorb the red light, right? So if the phytochrome absorbs red light, it is in this form, then it gets, in the presence of red light, right, it will get transformed into another form of phytochrome which is called PFR. That means the phytochrome that is ready to absorb far red light. This is the active form. Right? Now, if you have phytochrome in the PFR form, right, and then you expose it to far red light, then it will go back to the inactive form, right, to PR, right? So this is far red light. So this one absorbs red, and this one absorbs far red. So the wavelengths are different, right? And this phytochrome, right, PFR is the active form. That is important for germination. Like if you have seeds, all right, and you're trying to get them to germinate, if you shine red light, it germinates. Okay, if you shine far red light, it doesn't. If you first give it red light, then you followed it by far red light, it doesn't germinate. First give far red light, then give red, it germinates. So that book has an interesting discussion of all those different possibilities, and how people came to this conclusion. So PFR is the active form, and so PFR, that is the far red absorbing, is the active form. And the example is the germination of lettuce seeds. And this is discussed in the book. So lettuce seed germination is a typical photoreversible. Because if you give it red, it germinates, then you give it far red, it goes back. Okay. So the typical photoreversible response controlled by phytochrome. So this is helped by red light. Inhibited by far red light. Then both the phytochrome, chromophore, and protein undergo conformational change. Conformational changes in response to red light. Yeah, let's stop here for today. Okay, I will continue on Thursday.